All right, hello everybody. And so this lecture, we're going to be doing a part 2 of our transport lecture looking at specifically osmosis, which is part of a passive transport. So if you want to come and join along with me, we're going to be starting on page 15 of your note packet. And the last time we talked, we introduced 2 ways in which materials can enter and leave the cell. And so we introduced our types of transport, and there's 2 types. The first one is the active transport. Big thing is this is going against the concentration gradient. We're going from a low concentration to that of a high concentration. And because of the intermolecular forces of a lot of these molecules, they don't want to do that. They have natural repelling of each other. And so as a result, it's going to require energy, okay? Energy required. Whereas with passive, this is going with or along the concentration gradient. This is going from high to low, okay? And this is the one that we're going to be working on today is what are the different types of passive transport, and more importantly, what determines what passes through the membrane. So for passive transport, again, no energy required. And when I'm talking about this, I'm talking about ATP energy. We're going to talk about ATP in a lot more detail later on, but just know that anytime we're talking about ATP, we're speaking about cellular energy, and that is what everything in our body runs on, is that ATP. So there are 3 main types of diffusion: simple, facilitated, and osmosis. But regardless, all 3 of these move in a high to low direction. So if we kind of take a look, if you distinguish all of them, the first one, simple diffusion, is where it's just simply going through the membrane. So this is going to be anything that is small, anything that is uncharged that can do this, and oftentimes they need to be nonpolar as well. So examples of this would be things like gases, sometimes water, okay, non-charged molecules. Now, facilitated diffusion's a little bit different. Here, this is where we are going through a protein channel. So anytime we talk about facilitated diffusion, we're talking about an integral protein or a protein channel. Okay, now, same premise, goes from high to low, but generally these are going to be larger or have a charge to them possibly, okay? But generally, what's going to end up happening is these protein channels can be controlled. These can be opened by different kind of chemical signals to allow certain substances in. So for this example, here we have glucose going to the cell. So that means my insulin right here would need to attach to this membrane for it to open and allow that glucose to come in. Now, we know that the blood glucose levels in a person is a negative feedback. So as soon as those sugar levels go down, that insulin is taken off and the protein channel closes up. Now, the last one is, again, passive transport going from high to low, but with osmosis, we're only dealing with the movement of water only, okay? And that's the key thing here, and that's what we're going to be kind of trying to determine today, is what direction will the water move based on the solute concentration in and out of the cell. So, again, remember that when we talk about diffusion, especially simple diffusion, it's just going from that high to low concentration. And again, these are different types of molecules that don't have a charge, they're relatively small, and they just go through the cell membrane itself. Okay, again, driven by kinetic energy, and that energy is stored by the concentration gradient. And again, they all want to move until they reach their equilibrium. But remember, the particles still move, even though they reach equilibrium. So what do I mean by that? Well, let's say that this is our semi-permeable membrane. Here are some solutes we have more solutes on the left, and so that's going to come over here until it's equal. Now, once it's equal on both sides, they're still moving. So for every one that goes over, one's going to go in. So we have a net movement of 0, even though we have those particles moving. All right, and so again, we see these are small non-charged molecules move until they reach an equilibrium. But when it says they're still moving, I'm saying is when it's at this point where they're equal, you have them still moving in and out. It's just at a fashion in which you have similar number moving out and similar number moving in, resulting in a net equal amount. All right, now, for facilitated, again, the big thing that you want to kind of highlight here is this, okay? This is going through larger and then polar molecules, all right? So again, those with a slight charge to them use protein channels, all right? Those are the two main things here to help move across the cell membrane. Still going down, the concentration gradient, still going from high to low, okay? Now there's two different types that we kind of want to look at examples. The glucose one that we just talked about here, but aquaporins, okay? These are special protein channels that allow for water to moving pretty quickly into the cell. This is oftentimes found in your kidneys, where you have these protein channels called aquaporins that when signaled by, let's say, an antidiuretic hormone, it closes it up, preventing the water from being able to leave, controlling the amount of water that's in the blood as a form of negative feedback. Now, osmosis is a special type of facilitated diffusion, but osmosis is a little bit tricky because oftentimes when we think of simple diffusion, we're thinking of like the solutes, the particles, the scent particles that we released in the classroom moving from one end to the other. But when we're talking about osmosis, it is only the diffusion of water. So anytime we're talking about osmosis, we're looking at the movement of water and nothing else. But key thing is, water is still going from an area of high concentration to that of a low concentration. So there's still no energy needed. All right, so let's kind of put this into perspective here. So I have a U-shaped cell here, okay? Now, here I have my membrane, okay? So that's my selectively permeable membrane. And so this is going to allow only certain molecules through. This selective permeable membrane will only allow water through. All right, so we'll call this side A, and we'll call this side B. Now, these little circles, these represent solutes, right? These are substances dissolved in a solution. So when we see them, we can clearly see that on side A, so if we kind of compare side A and side B, I want to look at two things. I want to look at the solute concentration, and I also want to look at the water concentration, also known as the solvent, all right? And I want to look at that for both sides, side A and side B. Okay, so on side A, I can see I have 1, 2, 3, 4, 5, 6 solutes on one side, and over on B, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So here we have a higher concentration of these solutes on side B, but we have a lower concentration of solutes in side A. But one of the things that when you're looking at a picture like this, okay, what you want to end up doing is you want to visualize the water cell. So all around those solutes, you have water molecules. And because of water's polar nature, these molecules are going to interact with this water. But take a look here. Okay, I'm filling in those empty spaces with water molecules, but automatically you can kind of see that we definitely have one side that has more water than the other, okay? And so in this particular case, for my water on side A, it's going to be high. And my water on side B is going to be low. All right, so we have situation here where we have high and low for both the solutes and the solvent or the water. But remember, osmosis is just the movement of water. And so I'm concerned about these two things here. And we know that water goes from high to low. So in that case, the water should leave from side A and move to side B until it reaches equilibrium, which in this case is what happens. The water level moves up and continues to move up until it reaches equilibrium. Remember, these solutes do not move, all right? That's because the membrane is selectively permeable. So only the water is moving in that particular case. So let's explore this a little bit further here. So a couple of definitions we want to make sure to have down. So solute, again, is things that's being dissolved. So examples of this would be, salt, sugar, those kind of things. Solvent, obviously, is going to be what's doing the dissolving. So for most of our examples, that's going to be water. And the concentration is, again, just talking about how many particles or solutes in a particular given area. So let's take a look. Well, if we take a look here we have a situation where in a concentrated solution, we have high amounts of solute and low amounts of water. And over here, we have low amounts of solute and we have high amounts of water or the solvent in this particular case. Now, based on the concentration, we can get different types of environment. So these three terms represent, and put this down, the environment the cell is placed in. So when we talk about things like hypertonic, hypotonic, or isotonic, we're looking at a situation where it's talking about what is happening outside of the cell. So in a hypertonic situation, this is where you're going to end up having a higher concentration of solute outside of the cell. Hyper means higher. Now, hypotonic, hypo means low, right? And so hypotonic solution is where you have the cell placed in something that doesn't have much stuff dissolved in the water. This is oftentimes we call distilled water. This is water where it has every kind of mineral taken out of it. Where regular tap water you get out of the faucet has things like fluoride, chloride, different things mixed into it. However, when those are solutes still, and so when you take all those out and just have pure water, you have what's called a hypotonic situation because it's low amounts in solute. Now, iso means equal, and so this is where you're going to have equal concentration of solute on both sides. These are definitely highlight-worthy terms. Be able to distinguish the term based on talking about the concentration of the water supposed to do solute. So, again, what's the environment compared like to the cell? Cell always has smaller amounts of solute dissolved in the cytoplasm. This one, distilled water equals hypotonic water, okay? So anytime we talk about distilled water, we're talking about a hypotonic situation. Salt water, that's gonna be a hypertonic situation. All right, so let's kind of go through each of these together here. So first one is what we consider to be a hypertonic solution. This is a solution where you have, I know this is broken up a little bit, but this is going to have a greater than concentration of the cell, meaning that it has more solute outside the cell. So as a result, because there's more water in the cell per given area, the water is going to end up moving out of the cell, okay? And so what happens to the cell? The cell shrinks, okay? So this is like putting a cell in a very concentrated salt water. And so remember, hyper means higher solute. Hypertonic has higher amounts of solute on the outside, so the water is going to leave. Hypotonic. Now, in a hypotonic situation, remember, hypo means low, okay? This is where a situation in which outside of the cell you have less solute than inside. So this, again, this is like that distilled water we were talking about. And as a result, if we kind of look at this picture, we can see that there are more solutes in the cell than outside the cell. So as a result, the water is going to move into the cell. And that's going to cause the cell to grow inside. Now, sometimes the cell, if placed in a very, very distilled condition, can end up bursting. And that's what we call lysis, everybody, okay? Lysis is when the cell essentially bursts, causing it to rupture. Now, hypotonic, low amounts of solute on the outside, water enters the cell. They're supposed to go into the cell. The third one is isotonic. Again, when you think iso, think equal, okay? And so this is a situation where you have similar amounts of solutes inside the cell and outside of the cell. And so as a result, you get a net movement of water of 0. So the cell stays the same size as a result of it. So they're equal, so net movement is into and out of the cell. So those are 3 solutions for osmosis. Now, in plants, it works the same way. In a hypertonic, now these are 2 terms that you want to make sure to distinguish both because there are questions on the test about it. Plasmolysis deals with cells in a hypertonic solution. So remember, plant cells have this central vacuole inside of them right here. And those can have water move into and out of the cell depending on how much water is available. So everybody in here has seen plants, and if you, especially now being in the late fall and as dry as it has been, we see the plants look a little sad. That's because they're in the hypertonic state. Whenever you have less water, water is going to leave, and that's going to cause the central vacuole to shrink, and that's going to leave this plant looking sad. If we're going to look at a plant, this is where it would look sad and kind of droopy. That's because that water is not pushing against that cell wall, given it its rigidness. So this is going to be plasmolysis. Flaccid is where this is going to be in a regular isotonic type of event. Now, hypotonic, this is where you are having a turgid cell. This is where you're placing that cell in an area of DI water, and that causes the cell to basically expand and potentially burst and damage it. Okay. So, big idea with this is that water always moves from an area of more water to an area of less, going from high to low, okay? So water always moves to the hypertonic. Remember this, okay? No matter what, hypertonic, the water always moves to it, okay? So let's take a look at. So if you want to go ahead and get your osmosis practice homework sheet out, let's go ahead and do some practice problems with that. So I'm going to kind of do this with you here. Okay. All right, so in this first one right here, this first question is just kind of using your notes to try to figure out, to make sure you know what hypotonic, isotonic, and hypertonic is all together. But I want to do a practice problem with you. So let's take a look at this osmosis practice problem right here, okay? So, number one, what part of the cell is most responsible for diffusion? We can go ahead and answer this. It's the cell membrane. Remember, the cell membrane is what determines what goes into and out of the cell. So whenever I work with a question like this, I always want to know two things, the solute and the solvent. Okay, so again, for my beaker, which is the environment the cell's put in, we want to look at two things, the solute concentration. So let's say that, let's just say our solute in here is going to be salt. And my solvent is going to be water, okay? Same thing here. In my cell, it's going to be solute and then water solvent. So it gives us a prompt here. It says in the beaker, we end up having a 50% water. So my solvent is 50%. And then in the cell, my solvent is 10%. All right. So what I need to do is figure out the solute concentration. And so here, this is all out of 100%. So if I have 50% water, that means I'm going to have 50% salt. If I have 10% water in my cell, that means I'm going to have 90% salt. But again, so based on this, what type of solution environment is the cell placed in? So what is outside of the cell? So if we kind of take a look at my solute concentration, okay, in the cell, we have my solute, I'm going to put salt here, my salt is 90%, whereas on the outside, my salt is 50%. So automatically, what type of solution environment is the cell placed in? It's in a situation where it has lower amounts of solute than in the cell. So if it's lower means it's hypotonic. Lower is hypotonic. So outside of the cell is hypotonic. Now, that's everything outside of the cell, everybody, okay? So outside is hypertonic, but inside of the cell, though, if that is hypotonic, that means my cell is going to be hypotonic. And then my cell is going to be hypertonic because it has higher amounts of salt on the inside. And so here, what type of solution does the cell have? It's going to be opposite of the environment. It's going to be hypertonic, okay? And remember, water always moves to the hypertonic, but let's visualize that. So when I want to figure out what direction the water is moving, I'm going to do this right here, okay? I'm focusing on the water for both. And so in the cell, I can erase this here. So in the cell, we have 50%. And inside the cell, we have 10%. All right? And so, again, the cell always goes, oops, sorry. Let me flip the switch. Sorry, my cell has 10%, and my beaker has 50%. And so, 50 is higher than 10, and so what's going to happen? My water is going to go into the cell, increasing the size. Now, let's check to see if a hypotonic. Let's try a problem here, okay? So kind of